Have you ever just paused to think about everything happening inside you right now? This constant quiet buzz of activity keeping you going.
It's pretty incredible, isn't it? A whole world operating without us consciously directing it.
Exactly. And today we're diving deep into something absolutely central to that.
Mm.
The structure and function of the hematologic system.
Just to,
Your blood,
right, the body's internal transport system. It's much more than just red fluid.
definitely. Think of it like a superhighway inside you, moving everything essential around. Our guide for this is a chapter from Understanding Pathophysiology, the 7th edition by Huether and colleagues.
A solid resource.
Yeah. And our goal today is really to break down this complex system into, understandable pieces
Yeah.
so you can walk away feeling like you've got a good handle on it.
Making the connections clear.
Right. So let's unpack this system. We're talking oxygen delivery, waste removal, defense,
Mm.
even keeping your pH stable. It's just a marvel.
It truly is. And it's funny how we often take it for granted, but understanding blood, the what and why of it, is so fundamental. It connects to almost every other system in your body.
Okay, so let's start right at the beginning. What is blood? Like, fundamentally.
Well, technically, it's a specialized connective tissue, which sounds a bit odd because it's liquid, right?
Yeah.
But that's what it is. And in adults, we're talking about roughly 6 quarts, maybe 5.5 liters, constantly circulating.
Constantly doing all those jobs we mentioned.
About once. And it's basically made of 2 main parts. You've got the liquid part, plasma, and then all the cells floating in the plasma.
Okay, plasma. That's the majority, right?
Yeah.
Like, volume-wise.
Yeah, a bit over half, maybe 55%. And it's mostly water, like 91% water.
But that other 9% must be pretty important stuff.
Oh, absolutely critical. That's where you find all the dissolved solutes. And quick distinction here, sometimes you hear about serum. Serum is just plasma after you take out the proteins involved in clotting. Important for lab tests sometimes.
Got it. So those solutes, proteins are a big part of that, right?
Huge part. Plasma proteins. Most are made in the liver, but there's a key exception, antibodies. Those defense proteins actually come from plasma cells, a type of immune cell.
Okay, and these proteins aren't just hanging out. They have specific jobs.
Very specific jobs. Take albumin. It's the most common one. Makes up, what, like 57%?
Oh, wow.
And its main gig is maintaining osmotic pressure specifically, colloidal osmotic pressure or oncotic pressure.
Okay, oncotic pressure. What does that mean in simple terms?
Think of albumin molecules as being too big to easily leak out of your tiny blood vessels, the capillaries. So they stay inside and act like little water magnets, pulling fluid back into the bloodstream from the surrounding tissues.
So it keeps the fluid balance, right, between the blood and the tissue.
Exactly. It keeps water where it needs to be. And you see this clinically if someone has low albumin, maybe from liver disease or severe malnutrition, that magnet effect weakens.
Fluid leaks out.
Fluid leaks out into the tissues, causing swelling, edema, and at the same time, the blood volume can actually decrease. It's a very neat balancing act.
That makes sense. Okay, so albumin is the fluid magnet. What about the other proteins, globulins?
Right, globulins are the next group, about 38%. They do different things. Some are transporters, carrying things like lipids, HDLs, LDLs, or iron. Others are involved in clotting, like prothrombin. And then you have the gamma globulins.
Antibodies.
These are your antibodies, yeah, your immune defenders. And then there are others like fibrinogen, which is essential for forming blood clots, plus various regulatory proteins, enzymes, hormones. It's a real mix.
Don't forget the ions, like sodium,
The
potassium.
electrolytes, yeah. Crucial for nerve function, muscle function, keeping that osmotic balance, pH. They're doing a lot too, even though they're small.
Okay, so that's the plasma, the liquid matrix. Now, what about the cells floating in it?
Right, the cellular components. These are the real workhorses. You've got erythrocytes, leukocytes, and platelets, red cells, white cells, and platelets.
Let's start with red blood cells, erythrocytes. They're the most common.
By far, they make up almost half the blood volume in men, a bit less in women. We're talking millions per cubic millimeter.
And their main job is?
Oxygen transport, simple as that. They carry hemoglobin, which binds the oxygen.
And they have that unique shape, right? The biconcave disc.
Exactly. That shape is perfect for maximizing surface area for oxygen diffusion. But what's maybe even more amazing is their flexibility, their reversible deformability.
Okay, explain that.
Well, think about capillaries. Some are incredibly narrow, like 2 micrometers wide. A red blood cell is normally 6 to 8 micrometers wide.
So how does it fit?
It squeezes. It literally folds and deforms itself to pass through, then pops back into shape on the other side. The book has a figure, 22.2, showing this in the spleen. It's Wild. This flexibility is absolutely critical for getting oxygen everywhere.
Wow. But they don't live forever?
No, because they lack a nucleus, and most organelles, they can't repair themselves or divide. So their lifespan is limited, about 100 to 120 days, constant turnover.
Okay. Next up, leukocytes, white blood cells, the defenders.
Yep, your immune system's mobile units. Far fewer than red cells, but vital for fighting infection and cleaning up debris, like dead cells.
And there are different types. I remember granulocytes.
Right, two main groups. Granulocytes have these visible granules in their cytoplasm and weird lobed nuclei. They include neutrophils, eosinophils, and basophils. They're all phagocytes, meaning they can engulf stuff.
And they can move around.
Yeah, they can do this amoeboid movement and squeeze out of blood vessels. That's called diapedesis to get to sites of infection.
So neutrophils, what's their specialty?
They're the most numerous, like 65, 75%. They're the first responders, the main phagocytes in early inflammation. They gobble up bacteria and debris. Short lifespan, though, maybe a day or two in tissues.
Okay. Eosinophils.
Eosinophils are involved in fighting parasites and also play a big role in allergic reactions. Their granules have enzymes that can break down histamine, helping control inflammation, but they can also cause damage and allergies like asthma.
And basophils, the
Yeah.
rare ones.
Less than 1%. They're similar to mast cells found in tissues. They release histamine, heparin, and other chemicals involved in allergic responses and inflammation.
Okay, so those are the granulocytes. What's the other group?
Granulocytes, no obvious granules. This includes monocytes and lymphocytes.
Monocytes become macrophages, right?
Exactly. Monocytes circulate for a bit, then move into tissues where they mature into macrophages. These are powerful, long-lived phagocytes. Think of them as the heavy-duty cleanup crew. They have different names depending on the tissue. Kupffer cells in the liver, alveolar macrophages in the lungs, and so on. They form what's called the mononuclear phagocyte system, or MPS.
And lymphocytes, they're the immune specialists.
Precisely. T cells, B cells, plasma cells, they orchestrate the specific adaptive immune response. Then you also have natural killer cells, NK cells. They're lymphocyte-like, but they can kill tumor cells and virus-infected cells directly without needing prior exposure, kind of like immune system assassins.
Cool. Okay, last cell type, platelets or thrombocytes.
Right. And these aren't even technically full cells. They're small, irregular fragments of cytoplasm that bud off from huge cells in the bone marrow called megakaryocytes.
But they're crucial for clotting.
Absolutely essential. They stop bleeding. They're filled with granules containing stuff needed for coagulation. When there's an injury, they get activated, change shape. The book has figure 22.5 showing them getting these spiny projections, pseudopodia, and stick together to form a plug.
How long do they last?
They circulate for about 8 to 11 days, then they get removed by macrophages, mostly in the spleen.
Okay, speaking of the spleen, that brings us to the lymphoid organs. They're tied into this system too.
Deeply connected. The lymphoid organs are where many lymphocytes and those mononuclear phagocytes do their work. We divide them into primary and secondary organs.
Primary being.
Thymus and bone marrow, where lymphocytes are born and mature.
And secondary.
Spleen, lymph nodes, tonsils, Peyer's patches in the gut. These are where immune responses are usually initiated.
Okay, let's unpack the spleen a bit more. You said platelets and old red cells get removed there.
Yes, it's a major filter. It's about the size of your fist. It does several key things. Filters antigens from the blood, cleanses the blood using those macrophages we talked about, destroys old red blood cells, initiates immune responses, and it even acts as a blood reservoir.
A reservoir, how does that work?
It has distinct areas. The white pulp is lymphoid tissue full of T and B cells for immunity. The red pulp has these venous sinuses that can store quite a bit of blood, maybe up to 300 milliliters. If your blood pressure drops, your sympathetic nervous system can trigger the spleen to contract and squeeze about 200 milliliters of that stored blood back into circulation.
Wow, like a backup tank.
Pretty much. It also has a network of macrophages filtering the blood flowing through those sinuses. Figure 22.6 gives a sense of this architecture.
But you can live without a spleen.
You can, yes. It's not absolutely essential. But removing it, a splenectomy, has consequences. People might have higher white blood cell counts, lower iron levels, a weaker immune response to certain bacteria, especially those with capsules, and you might see more oddly shaped blood cells circulating because that quality control filter is gone.
Okay. What about lymph nodes? We feel those swell up sometimes.
Right. They're part of the lymphatic system, filtering lymph fluid, not blood directly. They're like little encapsulated filters scattered along your lymphatic vessels. Lymph flows in, percolates through sinuses packed with immune cells, and flows out. Figure 22.7 shows a cross-section.
So that swelling is the immune response happening.
Exactly. They're He sites for antigens, like bits of bacteria to meet lymphocytes. Macrophages inside also filter debris. If an infection is detected, B cells start multiplying like crazy in areas called germinal centers, causing the node to enlarge. It's a sign your immune system is actively fighting something.
Okay, so we know the components and where they hang out. How are all these blood cells actually made? Where does it start?
It all starts in the bone marrow. That's the primary factory.
Inside our bones.
Yep, within the cavities.
Hmm.
You have red marrow, which is the active blood-producing marrow, and yellow marrow, which is mostly fat and inactive, though it can convert back to red marrow if needed. In adults, red marrow is mainly in the flat bones, pelvis, vertebrae, skull, ribs, and the ends of the long bones, like the femur and humerus.
And it's not just empty space in there.
Oh no, it's a complex microenvironment. There are specific niches that support the stem cells. Two key types of stem cells live there: hematopoietic stem cells, HSCs, and mesenchymal stem cells, MSCs.
HSCs, those are the ones that make all the blood cells.
Exactly. They are the multipotent progenitors for all blood cell types. And the MSCs are stromal cells. They provide the support structure and can differentiate into bone cells, fat cells, cartilage cells, basically building the niche for the HSCs.
How do the finished blood cells get out?
They have to squeeze through tiny openings in the walls of the venous sinuses within the marrow that diapetesis process again to enter the circulation. It's a regulated exit.
And this whole process, differentiation, how does one stem cell decide to become, say, a red blood cell versus a neutrophil?
It's a fascinating cascade. All cells start from that single fertilized egg, undifferentiated. The HSCs in the marrow are still pluripotent to a degree they can self-renew, make more HSCs, or they can differentiate into progenitor cells. These progenitor cells become progressively more restricted.
So they commit to a lineage.
Right. They commit to either the lymphoid lineage making lymphocytes or the myeloid lineage, which gives rise to everything else: granulocytes, monocytes, red cells, platelets. Figure 22.9 in the book maps this out really well. It's guided by signals, primarily cytokines and growth factors, often called colony-stimulating factors or CSFs. They tell the cells which path to take and when to mature.
So there are different pools of cells.
Yeah, you can think of the bone marrow pool with the stem cells and maturing cells, and then the circulating pool with mature cells and some progenitors. Figure 22.8 shows this concept of hematopoiesis.
And the body can speed this up if needed, like after bleeding.
Absolutely. It can convert yellow marrow to red, make the stem cells divide faster, and push the daughter cells through differentiation more quickly. Production can ramp up significantly.
This production, hematopoiesis, it's happening constantly, right?
Non-stop. After birth, it's pretty much all in the bone marrow. You need to produce something like 100 billion new blood cells every single day just for maintenance.
100 billion. That's staggering.
It is, and that baseline increases dramatically in response to things like infection or chronic blood loss. Sometimes, if the marrow is really struggling, you might see extramedullary hematopoiesis blood cell production starting up again in the liver or spleen, where it happened before birth. But that usually signals an underlying disease process.
Let's zoom in on red blood cells erythropoiesis. How does that specific process work?
It's a stepwise maturation. An HSC commits down the myeloid line, becomes a progenitor, then a proerythroblast. This cell then goes through several stages called erythroblast or normoblast stages. During this time, it actively synthesizes hemoglobin and gradually shrinks and ejects its nucleus.
It loses its nucleus.
Yeah, to make more room for hemoglobin. The cell becomes a reticulocyte, basically an immature red blood cell that still has some residual RNA. These are released into the blood and mature into erythrocytes within about 24 hours. Figure 22.11 illustrates this nicely.
And the reticulocyte count, that tells you how fast the factory is running.
Exactly. Normally, about 1% of circulating red cells are reticulocytes. A higher count means the marrow is churning out new red cells faster, maybe in response to anemia or blood loss.
What controls this production rate? Is it just automatic?
No, it's tightly regulated primarily by the hormone erythropoietin, or EPO.
EPO, where does that come from?
Mostly from specialized cells in the kidneys. These cells sense oxygen levels in the blood flowing through them. If oxygen levels drop hypoxia, they release more EPO.
And EPO tells the bone marrow.
Make more red blood cells. Specifically, it stimulates the proliferation and differentiation of those proerythroblasts. More red cells mean more oxygen carrying capacity, which corrects the hypoxia, and then the kidney cells reduce EPO secretion. It's a classic negative feedback loop. Figure 22.60 shows this loop.
How much can EPO boost production?
A lot. In severe hypoxia, like at high altitude or with certain lung diseases, it can increase red cell production maybe sevenfold from around 2.5 million per second up to 17 million per second.
Wow. That's why synthetic EPO is used clinically, right?
Yes, recombinant human APO is a major treatment for anemia, especially in patients with chronic kidney disease who can't make their own EPO. A common side effect, though, is increased blood pressure.
Okay, so EPO drives production, but the red cells need to make hemoglobin. How does that happen?
Hemoglobin synthesis is happening inside those developing erythroblasts. Hemoglobin itself is a complex protein making up about 90% of the red cell's dry weight. Figure 22.12 shows its structure. It has 4 polypeptide chains called globins arranged in 2 pairs, and nestled within each globin chain is a heme group.
And heme contains iron.
Yes, each heme group is an iron protoporphyrin disk. That iron atom is the critical part for binding oxygen. It needs to be in the ferrous F2+ Fe state to bind O2. When oxygen binds, the iron briefly gets oxidized to Fe3+, that's oxyhemoglobin, but it gets reduced back when oxygen is released, becoming deoxyhemoglobin.
What if it stays oxidized?
If the iron gets stuck in the F3+ state, that's called methemoglobin, and it cannot bind oxygen effectively.
And one hemoglobin carries 4 oxygen molecules.
When fully saturated, yes. 4 heme groups, 4 oxygen binding sites. This increases the oxygen carrying capacity of blood about 100 times compared to just dissolving oxygen in plasma.
Does binding one oxygen affect the others?
Yes, it causes a conformational change in the hemoglobin molecule that actually increases the affinity of the remaining sites for oxygen, makes it easier to load up fully in the lungs.
What about other gases, like carbon monoxide?
Ah, CO. Big problem. Carbon monoxide binds to that same iron site in heme, but with an affinity about 200 times greater than oxygen. So even small amounts of CO can effectively block oxygen transport. Hemoglobin also binds carbon dioxide, but at a different site on the globin chains, not competing directly with oxygen. And interestingly, it also binds nitric oxide, NO, which helps regulate blood vessel dilation.
Making all this red cells, hemoglobin requires specific ingredients, right? Nutrients.
Absolutely. The textbook has a good table, 22.4, outlining them. You need protein for the globin chains, of course. Vitamins are crucial, especially B12, and folate for DNA synthesis, which is vital for rapidly dividing cells like erythroblasts. Deficiencies there cause megaloblastic or macrocytic anemia, where you get large, immature red cells.
B12 needs intrinsic factor from the stomach to be absorbed, right?
Correct. And folate deficiency is relatively common, especially important to prevent during pregnancy due to the risk of neural tube defects. Then you need vitamin C, E, B6, and minerals like iron, obviously for heme synthesis, and copper. Deficiencies lead to different types of anemia.
Okay, so we make them, they circulate for 100, 120 days.
Yeah.
Then what? How are old red cells removed?
As they age, they become less flexible, more fragile. Their membranes change. Macrophages, primarily in the spleen's red pulp, recognize these changes and engulf the senescent red cells. If the spleen is gone or overwhelmed, Kupffer cells in the liver take over.
And the hemoglobin gets broken down.
Yes, the globin chains are broken down into amino acids, which get recycled. The iron is carefully extracted from the heme and recycled too. The remaining part of the heme molecule is converted into biliverdin, then into unconjugated bilirubin.
Unconjugated, meaning not water-soluble.
Right. It travels through the blood, bound to albumin, to the liver. The liver cells take it up and conjugate it, attaching glucuronic acid, which makes it water-soluble. This conjugated bilirubin is then excreted into the bile, goes into the intestine.
And eventually leaves the body.
Mostly. Intestinal bacteria convert it to urobilinogen. Some of that is reabsorbed, but most is further converted and excreted in feces, giving stool its characteristic color. Figure 22.13 diagrams this whole bilirubin metabolism. If red cell destruction is excessive, the liver can get overwhelmed, leading to a buildup of unconjugated bilirubin jaundice and potentially bilirubin gallstones.
What about the iron that gets recycled? How does that work? The iron cycle.
The body guards iron very carefully. About 2/3 of your body's iron is in hemoglobin at any given time. Most of the rest, maybe 30%, is stored inside cells, primarily bound to a protein called ferritin. Some is stored as hemosiderin, which is less readily available. These stores are mainly in macrophages and liver cells.
So when old red cells are broken down, the macrophage saves the iron.
Yes. It releases the iron, which then binds to a transport protein called transferrin in the blood. Transferrin acts like a taxi, delivering the iron primarily back to the bone marrow, where developing erythroblasts grab it via transferrin receptors to make new hemoglobin. Figure 22.14 shows this cycle.
We don't absorb much iron from food.
We only absorb about 1 to 2 milligrams a day, but we need around 25 milligrams for daily hemoglobin production, so that recycling pathway is absolutely critical.
What controls this iron balance? Absorption, release from stores.
The key regulator is a hormone called hepcidin made by the liver. Hepcidin basically controls the doors that let iron out of cells, intestinal cells absorbing dietary iron, and macrophages releasing recycled iron. It does this by binding to the iron exporter protein, ferroportin, causing it to be degraded.
So high hepcidin means less iron gets into the blood.
Exactly. Less absorption, and iron gets trapped in storage within macrophages. Inflammation is a potent stimulus for hepcidin production. This explains the anemia of inflammation, or anemia of chronic disease. Inflammation triggers hepcidin, which restricts iron availability for erythropoiesis, even if body stores are adequate.
Fascinating. Okay, briefly, what about developing white blood cells and platelets?
Similar principles. Myelopoiesis covers the development of granulocytes and monocytes from those myeloid progenitors in the bone marrow. They mature and are released. Granulocytes also hang out in marginating pools, stuck to vessel walls, ready to deploy quickly. Monocytes head to tissues to become macrophages. Lymphopoiesis is lymphocyte development from lymphoid progenitors, often involving maturation in lymphoid organs outside the marrow. Platelet development, thrombopoiesis, is unique.
From those giant megakaryocytes.
Right. The megakaryocyte matures in the bone marrow, gets really big and complex, and then its cytoplasm basically fragments or buds off into thousands of individual platelets. No nucleus in the platelet, but they have granules and mitochondria.
What controls platelet production?
Primarily a hormone called thrombopoietin, or TPO, made mostly by the liver. TPO stimulates the megakaryocytes. Interestingly, TPO levels are regulated by existing platelets. If platelet counts are normal or high, platelets bind up TPO, so less reaches the marrow. If platelet counts are low, more TPO is free to stimulate production. It's another neat feedback loop. Inflammation can also boost TPO via IL-6.
Okay, so we have all these cells circulating, doing their jobs. What happens when there's an injury? How do we stop bleeding? That's hemostasis, right?
Exactly, hemostasis. It's the body's process to stop blood loss. It's a rapid, localized, and carefully controlled response involving three main components: the blood vessels themselves, platelets, and the plasma clotting factors.
What's the sequence of events, generally?
Okay, picture a small cut. First, the injured blood vessel immediately constricts vasoconstriction to reduce blood flow. Second, the injury exposes stuff beneath the vessel lining, like collagen. Platelets flowing by stick to this exposed area that's adhesion and become activated.
They change shape and release chemicals.
Right. They aggregate, sticking to each other, forming a temporary platelet plug. This is primary hemostasis. Third, the activated platelets and damaged tissue release factors that kick off the coagulation cascade involving those plasma clotting factors. This leads to the formation of fibrin.
Fibrin. That's the clot.
Fibrin strands form a meshwork over the platelet plug, trapping blood cells and making a much stronger, more stable clot. That's secondary hemostasis. Finally, the clot retracts, pulling the edges of the injury together, and eventually, controlled breakdown of the clot, fibrinolysis, begins as healing occurs.
So the blood vessels play an active role even before clotting.
Definitely. Normally, the healthy endothelium, the inner lining of blood vessels, actively prevents clotting. It produces things like nitric oxide and prostacyclin that keep platelets calm and vessels dilated. It also has molecules on its surface that inhibit coagulation.
But when it's damaged?
That barrier is broken. The underlying matrix is exposed. The endothelial cells themselves get activated, release von Willebrand factor, VWF, which is crucial for that initial platelet adhesion, and they also expose tissue factor, which jump-starts the extrinsic coagulation pathway.
And the platelets?
Yeah.
What exactly do they do besides form the plug?
They're central players. They adhere, aggregate, provide a surface for the coagulation reactions to happen efficiently, release factors that enhance vasoconstriction and clotting, and later release growth factors to help with tissue repair.
So having enough platelets is critical. Low platelets, thrombocytopenia.
Leads to bleeding problems. Below a certain level, maybe 100,000 per cubic millimeter, clotting takes longer. Below about 20,000, you can get spontaneous bleeding, like petechiae or purpura.
You mentioned platelet activation as a process. Adhesion, activation, aggregation.
Yeah, it's linked. 1. Adhesion. Platelets stick to the damaged site, mediated by VWF. 2. Activation. This triggers shape change, making them spiny, and degranulation. They release chemicals from their granules.
All kind of chemicals.
Things like ADP to recruit more platelets, serotonin for vasoconstriction, calcium, which is needed for clotting, and factors from alpha granules like fibrinogen, growth factors, and platelet factor 4, which helps neutralize heparin.
They also make something themselves, TXA2.
Right, thromboxane A2. They synthesize this, and it powerfully promotes more platelet aggregation and vasoconstriction. It directly counteracts the prostacyclin from healthy endothelium. 3. Aggregation. Stimulated by ADP and TXA2, platelets activate receptors that bind fibrinogen, linking platelets together. 4. Clotting system activation. The activated platelet surface provides the perfect scaffold for the clotting factors to assemble and work efficiently.
There was a note about sticky platelet syndrome.
Ah, yes, SPS. It's an inherited condition where platelets are just inherently more prone to aggregating, increasing the risk of clots, often triggered by stress, usually treated with low-dose aspirin.
So for minor cuts, this platelet plug might be enough?
Often, yes. It can seal small injuries in maybe 3 to 5 minutes, but for larger injuries, you need the reinforcement of the fibrin clot.
Which comes from the clotting factors, the coagulation cascade.
Right. These are mostly plasma proteins made by the liver that circulate in inactive forms. They get activated sequentially, like dominoes falling.
Intrinsic and extrinsic pathways.
Yes. The intrinsic pathway is activated by factors already within the blood contacting damaged vessel surfaces. The extrinsic pathway is triggered by tissue factor released from damaged cells outside the blood vessel itself.
But they lead to the same result.
They both converge on activating factor X. Once factor X is activated, it initiates the common pathway, which leads directly to the formation of thrombin.
And thrombin is the key enzyme.
Absolutely central. It converts fibrinogen, which is soluble, into fibrin, which is insoluble, and polymerizes to form the fibrin mesh. Thrombin also amplifies its own production and activates platelets, endothelial cells. It really drives clot formation. Figure 22.15 in the text shows this fibrin mesh trapping cells nicely.
But this cascade needs to be controlled, right? Otherwise, we'd be clotting all the time.
Precisely. There are several natural anticoagulant mechanisms. Antithrombin III, AT33, circulates and inhibits thrombin and other activated factors, especially when boosted by heparin-like molecules on the endothelial surface. Tissue factor pathway inhibitor, TFPI, blocks the extrinsic pathway. And then there's the protein C, protein S system.
How does that work?
Thrombomodulin on the endothelial surface binds thrombin, changing its function. This complex then activates protein C. Activated protein C, along with its cofactor protein S, degrades factors VA and ADA, which are crucial amplifiers in the cascade, so it dampens clot formation.
Deficiencies in these inhibitors would cause problems.
Yes, inherited deficiencies of AT33, protein C, or protein S lead to hypercoagulable states, a much higher risk of thrombosis. Inflammation can also tilt the balance towards clotting by reducing the activity of the protein C system.
Okay, so the clot forms, bleeding stops. Then what?
Two main things happen: clot retraction and clot lysis, fibrinolysis. Retraction happens fairly quickly. The trapped platelets contract, pulling the fibrin strands tighter, making the clot denser and pulling the wound edges closer. This usually happens within an hour or so.
And lysis, breaking it down.
Right, fibrinolysis. This is equally important to prevent clots from blocking vessels permanently. The key enzyme here is plasmin. It circulates as an inactive precursor, plasminogen.
How does plasminogen get activated?
Primarily by tissue plasminogen activator, TPA, which is released slowly from endothelial cells near the clot. TPA binds to fibrin within the clot, efficiently converting trapped plasminogen into plasmin right where it's needed. There's also urokinase-like plasminogen activator, UPA, which works more outside vessels. Figure 22.16 shows this system.
And plasma just chops up the fibrin.
Exactly. It degrades the fibrin mesh into smaller pieces called fibrin degradation products, or FDPs. These are then cleared from circulation.
Is that where D-dimer comes from?
Yes, D-dimer is a specific type of FDP, formed when cross-linked fibrin is broken down. Measuring elevated D-dimer levels in the blood is a very useful test to suggest that significant clotting and subsequent fibrinolysis has been occurring somewhere in the body, like in DVT or PE.
Okay, wow. That's a lot of intricate machinery. How does this system look different in kids or as we age? Let's start with pediatrics.
Blood cell counts are generally higher right at birth compared to adults, then they gradually decline during childhood. Platelets, interestingly, are usually at adult levels pretty early on. Table 22.7 summarizes some of these trends.
Why higher counts at birth, especially red cells?
Well, the environment inside the womb is relatively low in oxygen compared to breathing air. This hypoxia stimulates EPO production, leading to higher red cell counts, a state called polycythemia of the newborn. After birth, oxygen levels rise, EPO drops, and red cell production slows down. Reticulocyte counts are high initially but fall quickly.
Yeah.
Also, fetal red blood cells have a shorter lifespan than adult ones.
What about white blood cells in newborns?
They have them, but functionally they're immature, especially phagocytosis and some aspects of the specific immune response. So newborns are more vulnerable to certain infections initially. Their neutrophil and lymphocyte counts are high at birth, but change over the first few years.
But they get some protection from mom.
Yes, crucial passive immunity comes from maternal IgG antibodies crossing the placenta and more antibodies if they breastfeed. By about 2 months, their own immune system is usually mature enough to respond well to vaccinations, and then immunity builds up through exposures and vaccines over time.
Okay, now flipping to the other end of the spectrum, aging. What changes happen in the hematologic system as we get older?
Overall, blood composition stays remarkably stable throughout most of adult life. The changes with advanced age are often subtle but can be significant. Well, there can be a slightly increased risk of iron deficiency due to potentially reduced dietary intake or absorption. Red blood cell lifespan is usually normal, but the marrow might respond a bit more sluggishly to replenish cells after bleeding. The red cells themselves might become a little less deformable, slightly more fragile.
And the immune side, white blood cells.
Lymphocyte function tends to decline somewhat with age. T cell function, important for cellular immunity, can diminish. And the humoral system, involving B cells and antibodies, might be less responsive to new challenges, like novel infections or vaccinations.
What about clotting, platelets and factors?
Platelet counts might slightly decrease, but paradoxically, platelet adhesiveness sometimes increases. Also, levels of some clotting factors, like fibrinogen, factors V7, 9X, and von Willebrand factor, tend to increase with age.
So if we connect this to the bigger picture, these age-related changes, while maybe normal aging, could contribute to increased risk of things like infection or thrombosis in older adults.
That's generally the thinking, yes. They don't represent disease in themselves, usually, but they can lower the threshold or reserve capacity, potentially impacting overall health in response to stress or illness.
Wow, what an incredibly detailed system. Okay, let's try to wrap this up. Key takeaways from this deep dive.
I'd say first, just appreciating the complexity and dynamism. The hematologic system isn't static. It's constantly working, transporting, defending, repairing.
Right. And second, understanding the components. Plasma, with its vital proteins like albumin for fluid balance, and the specialized cells, oxygen-carrying red cells, diverse defensive white cells, and clotting platelets.
Third, the continuous production, hematopoiesis, primarily in the bone marrow, driven by stem cells and regulated by intricate signals like EPO and TPO.
Fourth, hemostasis, that amazing balance between stopping bleeding rapidly via vessel constriction, platelet plugs, and the coagulation cascade, versus the equally important control mechanisms and fibrinolysis to prevent unwanted clots.
And finally, recognizing that the system isn't the same throughout life. There are predictable developmental changes from infancy through old age that influence its function.
Okay, so here's a thought to leave everyone with. We've seen how precise and self-regulating this system is. What happens when just one small piece, one protein, one cell type, one regulator goes wrong?
Yeah, it really makes you think. How can understanding these fundamental mechanisms, this intricate balance, help us better grasp health, diagnose disease, or even develop new therapies, whether you're heading into healthcare or just want to understand your own body better?
Absolutely. Well, this has been a really packed, but I hope super insightful look at the hematologic system. Hopefully you feel a bit more confident navigating this crucial area.
I hope so too.
From the Last Minute Lecture team, thank you so much for joining us today. Keep diving deep into your learning.